Yeah.
Yes, I have just I have made you presenter now, so you should be able to share your screen.
Not yet.
Okay, let me try one more time.
Yep.
Okay.
Yes.
Not yet.
Can you see the whole screen?
Yeah, we can see it. Yeah, full screen. Yes. In our case, yes.
Okay.
Maybe
I can.
I think.
And I will talk on the analysis of microplastics by pyrolysis GC coupled to a high-resolution mass spectrometer. And while I'm Silvia Lacorte, and this work has been done with Ana Torres, who is doing the PhD and developing the methods with this technique. And we are from the Department of Environmental Chemistry in the Spanish Research Council Institute in Barcelona. So in here I put like a plastic toy and how we go from the plastic toy or from any plastic in the environment to a reliable spectra that can be used for identifying and also for quantifying microplastics. I will go a little bit fast because I think that we are running out of time. So basically we have seen in the previous presentation how to analyze microplastics, and I would like to highlight the challenges. First, it is in the sampling procedure, which has to be representative. Sometimes the samples are full of interferences, so often purification is required. But at the same time, as we do more manipulation of the samples, there is a higher chances of external contamination. We have also to take into account that we are analyzing polymers of different types. There are many types of polymers in the market and also in the environment. These polymers can age, so they change their properties, and this implies additional difficulties in the analytical analysis, and also that we are having different sizes, and the technique that is to be used needs to cope with these different sizes. So among the different techniques used in here, we are going to focus on pyrolysis GC-MS with Orbitrap mass spectrometer. And the objectives of the present work are to develop a method for the quantitative analysis of 10 microplastic polymers, to determine the quality parameters of the method, including the uncertainty, to validate the method by participating in an interlaboratory exercise on microplastics in water, because it is one of the biggest challenges to determine microplastics. Also to analyze microplastics and associated contaminants in air, and to perform non-target screening. So the work that I am going to present now has been published in Journal of Hazardous Materials. Here is the publication that provides all the specific information on the method developed. So one of the challenges is how to pre-concentrate the samples of the microplastics. So it can be water, it can be air. We are working in these two matrices mainly. There are different procedures that now I will not go into detail. But at the end, we finish with a filter, and in this filter, it contains all the microplastics that have to be characterized. So the procedure in here, it is something that needs to be optimized, but I will not go into detail into these aspects. So here we have our instrument. It's so we bought the Frontier pyrolyzer, and we coupled it to an Orbitrap, GC-Orbitrap MS. So at the end we have I would say a powerful instrument which is capable to determine microplastics, but also at high resolution. When we work with high resolution, the identification capabilities and also the quantification capabilities increase in a highly, because we are working with exact mass. So this is the procedure is laborious in any case, and it is expensive because everything of well, this instrumentation is very expensive, and therefore we have to work in conditions that the results that we obtain from this analysis are reproducible and quantitative. And this also one has to take into account when we started analyzing with this instrument of the capacity of the capsule. This capacity is 80, it can be 50 or 80 microliters. It's very small, so it means that everything in our analytical procedure has to be made small and without losing any of the plastics present in our samples. Also, it is important to work with instrumental blanks and method blanks, which I will expand this information after, and also to work in quality control conditions to prove the validity of the results. And this is challenging because at least at the time that we started working with microplastics, which is from 2017 when we started, there were no standards available. And therefore it was very difficult to do quality control analysis. So how we made it is we bought pure plastic materials and we drilled them with a diamond driller. And we have here different plastics, microplastics of the size of 10 to 100 micrometers. We weigh them in a ultra microbalance. And in here we have the different polymers and the different size ranges. And this allowed us to calibrate the system with accuracy. So I will talk of the different parameters on the pyrolysis GC-MS method. So to set up the method, we weigh 20 micrograms of each polymer standard directly into the pyrolysis cup. And in here we see a chromatogram, that's a standard. So you could expect 10 compounds, 10 peaks, but in real life, 10 compounds provide all these chromatogram full of hundreds of peaks. And therefore, the selectivity that of each polymer needs to be really optimized. So what we did in here, we have the 10 polymers, and we use deuterated polystyrene as an internal standard, which is very important to work with internal standard. It's this peak here. And when we extract the exact mass of each polymer, we see that all this background noise somehow disappears, and we see very neat peaks corresponding to each polymer. And here we see that this mass, the exact mass with 4 decimals, with an error lower than 5 ppm, it is very good to get this required specificity. Here we have the spectra of the polyethylene, which has been described before. It's like challenging to determine this compound, how we do it. So we have the chromatogram here, and we can click on any of these peaks, and we have a spectra that looks like that. It corresponds to the alkane chains, and then we have different possibilities of ions that we can determine, and always we get the one that has the highest intensity in reality and the highest specificity. And in this case, it's not one of the biggest peaks, but it is this compound in here, which is a C21. And we can check that by library confirmation. And therefore, it is the ion at MZ2082 that gives the highest specificity. For polycarbonate, another polymer, we see a very nice peak here, which corresponds to bisphenol. It is this peak here, but then we have the molecular mass in here. We have to think that many polymers, they produce the same ions. So therefore, it is very important to have specific ions corresponding to each polymer. So we have the molecular weight in, well, not the molecular, but this fragment in here. But and this is the one that we are using for quantifying this compound. In here we see the molecular weight at 228, which is very small, and then we have another fragment at exact mass that provides confirmation on the presence of polycarbonate polymers in the sample. And we did this for all the 10 polymers that we identified. In here, you cannot see this information, but it is in the paper. So we provide information on the 3 main ions at exact mass that are used for confirming the presence of a polymer in the sample. We go to the validation. The validation is done through the calibration, accuracy and precision, limits of detection, robustness, and stability. And with that, we do that with the standards, the ones we made, with the spike samples, with the reference materials, and in a interlaboratory exercise. The first challenge, though, it is calibration. So this is a peak of polypropylene, and this is a peak of polystyrene. In here, we have the calibration that in our case, it goes from 5 micrograms to 50 micrograms. We can see in the case of polystyrene that at 50, well, in reality at 40 micrograms, the peak is oversaturated. So it means that we have to go down in this line in here. And with our system, we cannot analyze concentrations lower than, we cannot weigh amounts lower than 5 micrograms. So it was good to hear that there are standards at a very low concentration, because we know that the compound is saturated, so therefore we cannot calibrate the system properly. And in the case of polypropylene in here, I want to show the curve quantified by external standard quantification and by internal standard quantification. By internal standard, using deuterated polystyrene, we get a very straight calibration with a very good coefficient of determination, and therefore this provides robust quantification of this polymer. When we go to the accuracy, we do it first with, for instance, this is an example of polyethylene, and we see that in here the green lights, what we have added in the cuvette, and what we are quantifying, it is very accurate. And also we did the same with the spiked bottled water. And when we analyze this filter, we find the recovery is close to 100%. So again, we have a good accuracy in the instrumental part, but also in the extraction part. When we go to the precision, intra and interday precision, this is a standard. This is a chromatogram corresponding to 5 micrograms of deuterated polystyrene. And we see that we have the response varies along the different injections. And this variation within days, it's of 16%. And within the same day, it's 10%. So in reality, this is quite a lot for an MS that we would like to have a precision of low than ideally 10%. But we see that this precision for the intraday, for the rest of the polymers, sometimes it can be very good, 3%, but sometimes it's really high, like 28%. So this variation provides uncertainty in the analysis. If we can correct this with the internal standards, of course, that gives a plus in the quantification accuracy. We go to the instrumental detection limits. The case of PMMA, there is no noise, so the limit of detection could be really, really low, but we take 1000 of intensity as an instrumental detection limit, because as you can see here, at the exact mass of this compound, there is no noise. So the instrumental could detect 0.02 micrograms absolute, which is very low. For the PET, yeah, we can see here in this chromatogram that there is some noise. So at the end, we use the conventional signal-to-noise ratio of 10 to calculate the instrumental detection limits. When we calculate the method, of course, it depends on the samples that we are extracting. This is an example, a chromatogram of an air sample. We see these two bumps, which is unresolved complex matrices, and the complexity of the chromatogram. It is very obvious, so it's quite challenging. And in here, it confirms the need to work at accurate mass to determine the different polymers with certainty. So method detections are difficult, I would say, difficult or challenging to calculate because of instrumental blanks, because problems in the extraction blanks, the type of matrix influences the method detection limits. Sometimes the recoveries are not calculated, or calculating the recoveries is not very straightforward. And it is also very difficult to spike environmental matrices at low concentration, basically because in general of the lack of standards. Here I would like to present the results because we had to battle quite a lot on how to get clean chromatograms. So this is to highlight the importance of carrying out blank analysis before analyzing microplastics, because otherwise it provides a lot of uncertainty and a lot of bias on where microplastics come from. So this is a blank chromatogram of a cup which was cleaned, roughly cleaned with a torch flame and methanol. But still there is a lot of interferences in here, as you can see. And in here we have a protocol for cleaning the cup to get an empty cup, which provides like a, you can see in here, no interferences from only the cup used to put the sample inside. And the same happens with a glass glass fiber filters, or the filter that it is used to put inside the cup. This is a glass fiber filter with no treatment, and here it is muffled at 450 degrees during 12 hours. That we can see that all these interferences are eliminated. So therefore, it is very important to muffle the glass and to use and to do a thorough cleaning of the pyrolysis cup. And in here we see at the end that working with like optimized cleaning procedures, the reproducibility of the PMMA polymer is very good. So at the end, this provides certainty in the analysis. We calculated the robustness and the uncertainty of the analysis, and we saw that in here, in blue, is the repeatability uncertainty. In red is reproducibility, that means injecting in different days. And in green is uncertainty during the procedure of extraction and weighing the microplastics. So altogether, we get uncertainties that in the best of the cases go to 15%, and in the worst of the cases it goes to 53%. The one which we have lowest and highest uncertainty is polyethylene, which polyethylene is also one of the microplastics that it is most difficult to identify, because the peak, the ion at MZ82 provides very low sensitivity, although specificity. We participated in a proficiency test for drinking water. This was not done with Orbitrap, but with pyrolysis MS with a single quad. And in here we see the results and the different laboratories participating. So in here it has to be indicated that most of the laboratories use FTIR instead of pyrolysis. That was done in 22. And only 1/3 or less of the laboratories were using pyrolysis. In here we have polypropylene, polycarbonate, polyvinyl chloride, and the total of polymers analyzed. And so our results is in here. So they were from 50 to 299 micrometers. And we provide green results, so accurate results for polypropylene and PVC. and for polycarbonate, we have some bias. But still, it was, well, we were quite satisfied because it's also the challenge to determine the 3 polymers and determine them with accuracy. So shortly I will present results on the analysis of microplastics in air. So we sampled with these pumps, we sampled 10 cubic meters, we analyzed with the pyrolysis, and we do the data analysis with the trace finder. So we analyze indoor and outdoor samples in department stores, in gyms as an example. This is a type of chromatogram that again we can see a lot of interferences. This is done at a resolution of 60,000. And besides, we also use the regions of interest multivariate curve resolution to process the data. So what we do in here, we have the retention time of the chromatogram and the MZ values, and we convert them and we export them to MATLAB. And with applying the regions of interest multivariate curve resolution, at the end what we get is chromatograms like this, that we have all the traces of all the MZs along the chromatogram. And from here we can identify the spectra and the retention time of the different compounds. And first we do that with the standards and then with the samples. What is the advantage of this method? That we can identify and quantify the compounds that for which we have standards, which could be the confidence level 1. In here, for instance, we have identified nylon, polypropylene, polystyrene, and the nitrile butadiene rubber, and we can quantify them with a frontier system, but also with a MATLAB. But then we go to the other compounds that might be present in the sample. That's the reason basically why we analyze high resolution. So you can detect your contaminants for which you have standards, but you can also identify other compounds which might be in the sample, providing like a much larger amount of information on the polymers, the additives in these polymers, and also associated contaminants. So in here, for instance, we have at confidence level 2. So these are compounds that we can search with a library search. That we have the retention index, the MZs, we have the spectra, and then we can identify them as, for instance, this list in here with a very low variance. This indicates in the samples that there have been identified, and this is a description of the different compounds identified. At confidence level 3, which it means that it's a tentative candidate, we have phthalates that, of course, if we have the standard of phthalates, we can identify them by retention time. With electronic ionization, we always get the ion at MZ149. So therefore, we need the retention time to identify the different phthalates. But in here, we can see that the elution profile of the different phthalates in the different samples, and also the retention time. We see several phthalates in these chromatograms, and the phthalates would be one of the plasticizers that are used to make the different polymers, especially PVC. At confidence level 3, we provide tentative candidates for which we cannot discern, for instance, structural isomers. For instance, in here we detected 2,4-aldehyde, but without knowing if it is 2 or 3, we cannot identify that because we did not have standards. But looking at the bibliography, we can see that 2,4-aldehyde is a solvent, a colorant, an adhesive, and therefore it has high chances to be detected in the samples associated to plastics. And at confidence level 4, we have a molecular formula, and we cannot identify what it is. We know that these are the potential candidates, and further research should be done. And I don't know if I have more time. Well, I continue.
How long do you need?
How long?
How many?
2 minutes. 2 minutes.
Yeah, sure, no problem. Yeah, please go ahead.
Okay, we do indoor air in schools, And this is a microplastic concentration in pink, well, in purple, it's indoor, and in yellow is outdoor. What we find is that indoor air, systematically, the concentrations are higher than outdoors. The concentrations range from 0.2 to 71 nanograms per cubic meter. And we can also identify in these graphs here on the right, the different polymers that behave in a different way in the different periods of the year, and also depending if it is indoor or outdoor. And this is very much related to the polymer and the sources. For instance, polystyrene, it's more related to textiles. So in winter, indoors, the concentrations are higher than outdoors. So the take-home messages is that the analysis of microplastics is not straightforward and a step-by-step optimization is required, that the quality control analysis is needed to ensure the reliability of the results, that the standards are needed, that the high-resolution MS permits a very specific identification of the polymers and also by doing non-target analysis of the associated contaminants. It is important to calculate the recoveries now that there are standards, which in many papers it is not done, and also that the external contamination should be checked to avoid false positives. And I would like to thank you for your attention, only to show this is a bus air picture and this is a house air. So in here again, it is the ability to analyze all what is present in these samples. And well, thank you again.
Thank you. Thank you.